slideshow. Okay, cool.
Okay, hold on one second. We'll wait for 1 minute. Okay, so as I noted, some of the discussion was a perfect segue into the next talk. So the next talk is by Philippa Johnstone, who is a recent graduate of the University of Arizona in my lab. She obtained her Master's of Science in Natural Resources with an emphasis on ecology, management, and restoration of rangeland ecosystems. Her research focuses on the use of seed balls in revegetation, as well as the germination behavior of different species and populations of milkweed. Her professional background includes conservation horticulture, native landscaping, and horticultural therapy. She's passionate about enhancing seed-based revegetation methods and including people with diverse strengths and abilities in the field of ecological restoration. Welcome, Philippa.
Thank you. So I don't need to do this slide. I'm just going to go right into it. So I always start presentations now with this slide because I think it perfectly captures not just graduate research, but just like being in the field that we're in, the business of seeds. It's deceiving because, when we have like these memories of being in preschool, or even like if you were lucky to get to work in a garden when you were younger, seeds always involved like a very simple procedure of putting them in water, putting them in a paper towel, Ziploc bag, bing, bang, boom. And I naively thought that that was going to be my experience with my second chapter, working with milkweed seeds. Turns out that's not the case. And that is because milkweed seeds, like many of the seeds that come from native plants and even edible crops, are very complicated. And they have a fascinating suite of traits and behaviors. And so today I will be talking about milkweed, but a lot of this applies to native plants in general and many of the edible crops that you might be working with as well. So seeds are wonderful. They're complicated. They drive me crazy, but I'm here to share my experiences with it, and hopefully some of this is transferable to what you're working with as well. So the focus of this talk will be on milkweed. And as it was really well highlighted in our last talk, this is considered a high value pollen and nectar resource for monarch butterflies, which are now classified as endangered. They're also really great resources for other pollinators too. And I think that's important to note because when we talk about pollinator restoration, the monarch butterfly is like our panda bear, like our poster child. But there's so many other insects in decline that really depend on this plant as well, or utilize this plant. So we're all kind of all hands on deck when it comes to pollinator restoration and specifically getting more milkweed in the ground. So there's a lot more demand for this plant. Despite the wide array of milkweed species that are native to the US and the high demand for growing them, there's still a disappointingly low number of species that are actually available for purchase or even for acquiring seed. And if you take that even a step further, if you're starting from scratch and you're growing from seed, a lot of the science research that was done on milkweed in this country, primarily, Western science, of course, and under the guise of like conventional agriculture, milkweed was viewed as a weed and it was a nuisance crop for a very long time. And so a lot of the research still to this day talks about getting rid of milkweed rather than planting it, leaving us kind of to fumble around and kind of, we have a lot of catching up to do. If you were to go and purchase milkweed from a nursery, I don't want to scare people away from doing that, but there have been a few recent studies. One of them was supported by the Xerces Society, found that there are harmful levels of pesticide residue on milkweed plants in nurseries. This could be intentional or unintentional due to drift. So this kind of pushes us even more to like really pay attention to milkweed and get more seeds and varieties out there, which leads me to the importance of growing milkweed from seed, because I think being in the seed library webinar today and understanding the value of storing seed and getting seed distributed, I think it's really important to focus on that, because you can kind of control the environment that the milkweed is growing in, hopefully, and have a little more say in where it's placed and where it's planted. So if you are growing milkweed from seed, you might have already encountered this or you might have heard of this, but oftentimes people encounter low or very highly variable germination success. This can occur from species to species. It can occur even from year to year. And also there's a lot of variation that can happen within a species. And so the information that you might find online for grower recommendations could actually be a little misleading, unintentionally, it could be biased towards one specific population or one species and not necessarily be relevant to the milkweeds that you're working with. Oops. Okay. Why is that? Well, milkweed, like many native plants, have been able to spread their native range and adapt to a variety of different environments. And part of that adaptation involves figuring out when to grow and how to grow, right? So, what influences germination behavior in a plant could include the plant's life history. A plant that's growing in a place where there is severe frost may want to avoid germinating during that time period or right before that time period. There could be a lot of variation or environmental heterogeneity in an area. So, wildfire occurrences, unpredictable rainfall that can also influence the behavior, the germination behavior of a plant. And of course, many plants have the ability to adapt in real time according to what they're experiencing in the environment. And so those maternal plants may pass on specific genetic traits based on what they experienced to their progeny, to their next generation. And then lastly, genetic flow between plant populations can also influence germination behavior, especially if we think about seeds that are wind dispersed and pollinated from flying insects or from wind. Genetic traits from one seed can pass on to the next plant and vice versa. And so this all kind of contributes to this high variation in germination behavior of milkweed. And thankfully, ecologists are catching on to this and they're looking at milkweed. So we're not completely starting from scratch here. There was a study that found 3 different species of milkweed, common, world, and swamp milkweed, had germination behavioral differences according to the population latitude. So if we were to look at this graph here, you can see common milkweed, Asclepias syriaca, and on the y-axis, you see the germination percentage, so 0 to 100%, excuse me. And then on the x-axis, temperature in Celsius. So each of these lines represents a different population. This dotted line is a northern population, and the dash line, and then the dotted line is the central population or southern population. Straight line is the central. And right off the bat, just looking at the distance between those lines, you can see that the northern population had a higher germination response to those increasing temperatures than these southern and central. A similar pattern was found in world milkweed, and then the opposite was found to be the case for swamp milkweed. So again, we're starting to pick up on these trends and variation in milkweed. And that's partially due to temperature sensitivity. So that's one of the things that we're looking at when we're talking about germination behavior. For any given plant, there's typically like a germination threshold within which that plant will germinate. When we're talking about temperature, there's going to be a minimum and a maximum temperature. And again, this is common with all plants, not just milkweed. And specifically for milkweed, a lot of the formal studies that have been done haven't necessarily looked at that temperature threshold. So oftentimes studies will have just looked at one temperature ranging anywhere between 7 to 30 degrees Celsius, which leaves us kind of scratching our heads like, okay, where in that ballpark do I need to germinate these milkweed? So our understanding of those temperature windows for germination is still pretty limited. So we want to understand how wide those temperature windows are for milkweed and what is that variation between species. So this leads me to my first research question, which is what is the germination response to temperature of non-dormant milkweed species and populations. I put non-dormant there because that is how I split up the milkweed. And that's because some milkweed actually have what we call seed dormancy. And I'm sure everyone on this chat has encountered seed dormancy, so I don't want to um, drill this into your head for the millionth time, but just to make sure that we're on the same page, I will just briefly explain what that means. So dormancy basically just means any type of germination inhibiting mechanism that a seed has imposed. That could be from the outer layer, the seed coat, or that can come from within, such as like certain hormonal levels within the chemistry of the seed. And so this is a genius way for plants to germinate their seeds at the right time, because for a lot of ecosystems, not all, but a lot of ecosystems, germination year round is not necessarily an great survival tactic. Because you might have danger of frost, or there could be other environmental variables that limit plant growth. And so dormancy prevents seeds from germinating until certain conditions are met. And the seed kind of knows, okay, it's showtime. I'm safe. Risk of mortality is low. I'm ready to germinate. So over half of our wild plant species have dormancy. Now, within milkweed, it's common that milkweed germinate during springtime. That's kind of pretty normal for a lot of forb species in the US. And what they have is a type 2 non-deep physiological dormancy, which essentially means they need to undergo some period of cold, moist conditions, like a wet winter, before followed by a warm temperature before they're able to germinate. However, not all milkweeds have dormancy. Some species actually have dormant and non-dormant populations. And then some species may have only dormant and only non-dormant. So it can get a little tricky when you're thinking about germinating your seeds and being successful. So a tactic that a lot of horticulturists have used, many of you have probably used before if you work with seed banks or seed libraries, is cold stratification. And this is basically us telling the seed, like, I tricked you. You just went through winter, and so now it's your turn to germinate. And so typically what that involves is taking the seeds, putting them in some type of medium, whether that's like vermiculite, some people do sand. I do paper towels, and I'll explain why like later in the talk. Moisten the medium, put them in a Ziploc bag or some type of container, and usually stick them in the fridge for a certain period of time. And then after this time elapsed, you then remove those seeds and you place them in a warm condition, like a spring, simulated spring, and that typically breaks dormancy and helps seeds germinate. However, as I mentioned a little earlier, dormancy is not consistent across milkweed species or even within species. So a study that was done by the Institute for Applied Ecology, looking at several populations of showy milkweed, Asclepias speciosa, sourced from throughout the Western United States, found that cold stratification does not necessarily enhance germination for all populations. So up here we have on the y-axis, again, this is germination percent going from 0 to 100. On the x-axis, you just have the population where those seeds were sourced from in the wild. So for example, somewhere in Washington, we've got a couple places in Oregon. These bars are different colors because, of course, you have a control, which is the clear bar. So no cold stratification. Those seeds just went in cold turkey. And then these other bars are different time periods of cold stratification, so ranging from 2 to 6 weeks. So for these populations, it's clear that there is some level of dormancy because you see a drastic increase in germination after cold stratifying those seeds. However, for these populations down here, such as the New Mexico populations, a Utah population, cold stratification did not seem to significantly enhance germination, implying that those seeds did not have the same dormancy that these guys did. Leading me to my next research question, which is, what is the germination response to cold stratification and temperature of dormant milkweed species and populations. So first, I'm going to talk a little bit about how I conducted the research, and then we're going to go into those results. Hopefully, I'm not going over time. We'll see. So methods include sourcing the seeds, and I will talk a little bit about this at the end of recommended seed sources. But essentially, I selected as many milkweed species as I could, not necessarily just confined to the Southwest where the study was done, but throughout the US. And then within those species, I tried to acquire populations that represented their native range as much as possible. An example here is Asclepias asperula, antelope horn. And so these black dots are areas where I was able to source those seeds, both wild harvested and commercially grown. And of course, controlling for seed age. This is not a huge issue for this particular research project, but in other settings, if you're working with pretty old seed, like 10 years and higher, you might actually start to see their germination decrease or even change because of the effect of long-term storage on seeds. That was not the case here. The seeds were all roughly the same age with it, give or take a few years. So that was like a important factor. And then doing the actual germination testing. So I'm not sure if y'all can see the video, but this is just showing you what a germination chamber looks like. It's essentially a giant box that you can put your seeds in, and it controls light, humidity, and temperature. So the seeds went into this chamber. They were watered daily just to keep moisture consistent. And then they were exposed to a cycle of 12 hours of light and dark, as well as a cycling of cool and warm temperatures. This is all to mimic as much as possible natural conditions, which is highly recommended for anyone who's doing germination testing. You want to basically mimic nature as much as possible by doing those diurnal shifts of light, dark, cool, warm. And for seeds that were not dormant, they did not undergo any cold stratification. They went into two different temperatures, a 20 to 30 degrees Celsius and a 25 to 35 degrees Celsius regime for 14 days. These temperatures are considered to be on the slightly warmer side of that graph that I was showing you earlier of a temperature window. And then, of course, the seeds that were dormant, same exact thing, except before going into the chamber, they were stratified, cold, wet stratified for 6 weeks. What I measured at the conclusion of each trial was the germination percent, really basic calculation. It's the total number of seeds that germinated divided by the total number of viable seeds. For any given batch of seeds, not all seeds are viable, meaning not all seeds are actually capable of germinating because they're either dead, the embryo is missing, or they were infected by some type of fungus or bacteria. If any of you have ever wild harvested seeds, sometimes you'll notice bugs will actually get inside the seed and just eat it from inside out. Other things can happen in nature, mutations. So even in a farm setting, you're not going to get 100% viable seed. So that is why germination studies typically only look at the viable seed rather than the entire seed population, if that makes sense. And I'll talk a little bit about seed viability later if there's time. Okay, so looking at our first question, which was investigating the germination response to temperature of those non-dormant seeds. And for the purpose of clarification, I classified non-dormant seeds as any seeds that had less than or equal to 30% dormancy. So here are some of the results. I'm not going to like walk you through every single one because that's just too much. But this graph is basically showing an overview of each of those species that had non-dormant populations. So I'm just going to move this down here. So on these panels, these are the titles of the species. Again, we've got our Asclepias asperula antelope horn all the way down to our world milkweed, Asclepias verticulata. Down here on the x-axis, we have our state codes. So these are where those seeds were harvested from. And then on the y-axis, germination percentage as well. So the two colors are representing those two different temperatures, 20 to 30 degree and 25 to 35 degrees Celsius, which are considered pretty warm temperatures to germinate in. Thankfully for us growers out there and restoration practitioners who are using seeds, for these specific populations within those species, we did not see any significant differences in germination response to temperature. So, all that to say that across the board, we saw pretty high germination. So 70% or higher seeds germinating in both temperatures. And to kind of thread this into what else is happening outside of my research, other studies, excuse me, have been done with similar species, sorry, the same species. So Asclepias asperula, subulata, which is rush milkweed, and subverticulata, horsetail milkweed. There was also a study done by the Institute of Applied Ecology that found for their populations, they were even germinating in 15 to 25 degrees Celsius, which is a slightly cooler temperature range. Also, they found no dormancy in those particular populations, in addition to Asclepias erosa and rush milkweed as well, subulata. Oh goodness, I'm sorry, that shouldn't be like that. Um, so a lot of what ecologists are interested in are finding patterns in in any possible way if they do exist in nature. So one of the ongoing theories about germination behavior is that there might be some climate-related patterns going on here. For these particular species, these particular populations, these came from more arid climates. And so there's this theory, this ongoing theory that aridity might play an influence in how a seed's germination behavior is developed. So for example, seeds with wider germination temperature windows in other studies that came from more arid regions may have been actually taking advantage of rainfall in climates where the rainfall was either very low or unpredictable. So what might be happening is the plant has adapted to be able to take advantage of that rainfall and not kind of be fussy about the temperature. So basically saying, okay, it's raining, I'm going to take advantage of this and I'm going to grow. Now, that's not always the case, but there are some studies, including milkweed, that have found that this is actually starting to support that theory. So to answer the question earlier about the germination response to temperature of those non-dormant species and populations, again, we found similar germination rates in both temperatures as well as reaching optimum or higher germination. Next steps or further questions, because research always just leads to more questions, right? Is what is that germination response going to look like for the 15 to 25 degrees Celsius? And based on that other study, I would guess that those same species would also do well in a lower temperature, giving them a wider germination window. And again, looking for those patterns in nature, perhaps linking climate factors such as aridity to help explain this wider germination temperature window. So now we're going to move into the second question, which was about dormant seeds. So what is their response to cold stratification and temperature? And again, dormant seeds, just for clarification, were any populations that had greater than 30% of their seeds dormant. So the results here, this graph is a little more patchworky. If you recall that earlier slide with the chili falling on the floor and spilling out of the pot, this is one of those situations where I encountered a lot of trial and error. And part of that was due to cold stratification issues. And so I'm missing a little bit of data still that I'm working to get to redo. So you'll have to work with me here. But what I am able to present to you are results that I was able to gather and to summarize. So for these particular species, these were the ones that had been cold stratified for 6 weeks and then went through those two temperature regimes. And what we're starting to notice with these dormant populations is that we did find significant differences between populations within the same species. So for example, in the antelope horn milkweed, the Asclepias asperula, you can see way down here, our Texas population had very low germination in both temperatures compared to our Nevada and our Utah populations. The Asclepias syriaca, the common milkweed, different germination responses as well, although similar in response to temperature, but across populations, we saw some differences, for example, between the Vermont population and the Minnesota population. In Asclepias viridis, the green milkweed, again, similar to the the common milkweed, we saw differences between populations, but also a potential preference for this cooler temperature. So my next step would then be to take it even further and look at whether Asclepias viridis can germinate in 15 to 25 degrees Celsius. Perhaps there's a narrower germination window for that species. I just thought it was interesting also to point out, I don't know if I can go backwards. I can. Okay. If you recall earlier that we had the same species with these populations doing reaching optimum germination without being cold stratified, to then these populations down here at actually higher elevations as well that required cold stratification to germinate, to get above 50% germination. So these differences in response to temperature may actually be contributing to this wider theory that plants that have timed their germination in early spring may have a competitive advantage over later emerging individuals, especially if you consider plants growing in highly densely vegetated areas like prairie ecosystems. So, for example, the Asclepias asperula and Asclepias viridis that we were working with, perhaps germinating in those even cooler temperatures, indicating that it's early spring is a competitive advantage for them to kind of take advantage of sunlight, take advantage of that rain, and get up there and send out those first leaves before anyone else. The Asclepias syriaca might be a slightly different story. So one of the interesting things that I found that sort of contributes to a couple of other studies specifically for this particular species is that northern latitude populations of common milkweed tend to have a wider germination window or niche breadth than lower latitude populations or southern latitudes. A theory attributed to that is perhaps populations that are growing in those northern latitudes deal with a lot more variability as to when that last frost date might be occurring. Oftentimes, it can occur even as late as May. And so that for that particular population, for those populations, it might be more advantageous to be able to spread out germination to kind of reduce the risk and or spread the risk a little bit more and be prepared to germinate later rather than timing germination specifically to that early spring in case there's another frost happening after. So some key takeaways from that particular aspect of the study that I didn't really show this graph. I didn't want to like overwhelm the slides and also just I didn't want to lose out on getting to questions and some of the other good tidbits. So what I wanted to show you, but I didn't have time to show you, is that for nearly all of these dormant populations, they responded positively to cold stratification. So that 6-week period of cold stratification enhanced their germination, but it was temperature dependent because as we saw earlier, not all populations did well in both temperatures and some populations didn't even reach 50% germination in either temperature. So next steps or questions for further research would, again, similar to the other to the other section of this research, is looking at that even cooler temperature, the 15 to 25, and will those populations that had low germination actually do better in the 15 to 25 degrees Celsius. And again, looking for those patterns, does the last frost date seem to have an effect on that germination window for some of those northern latitudes? And so putting this all together, I wanted to kind of, we really like hyper focused and zoomed in on the milkweed, and we learned a lot from this research. So I want to kind of zoom back out and bring this back to the general topic of seed libraries and just trying to be more successful when we're germinating these seeds, because the end goal, of course, with a seed library is to grow the plants, right? So the next couple slides are really just going to be about my recommendations for germination, for increasing your germination success. And this is like probably said over and over again, but knowing where your seeds come from really does matter. For native plants especially, I recommend using this USDA seed zone transfer map. It looks very psychedelic, lots of colors going on. But essentially, this map is made to kind of help you figure out where you're safe when you're sourcing your seeds. So how far away can you go when you're doing your sourcing? If you're down in Tucson, Arizona, perhaps I shouldn't be sourcing my milkweed seed from Minnesota or even in Texas. We have completely different climates, and so it's really important to factor that in. And thankfully, the USDA has poured a bunch of data into this and really looked at it to help people figure out where they are on that map. And then, of course, making sure your seeds have decent quality. So you're not always guaranteed good viability with wild harvested seeds. Things happen in nature. A drought could occur. The maternal plant could abort their embryos. And so you'll just get empty seed casings with no embryo. Or again, granivory. Some insects can get into those seeds and eat those seeds from the inside out. So it's really important to understand how viable your seeds are. It's a great starting point, and it can save you time and effort when you are germinating your seeds. Also, as we learned with this research, it's important to know if your seeds are dormant or not. You don't necessarily have to cold stratify all of your seeds, but if you are getting dormant seeds, it's important to go through those protocols of cold wet stratification in order to enhance your germination. If you're getting seeds from like Billy Bob Joe all the way up in Oregon, who I don't know, has a Ziploc bag of milkweed and has no other information for you, don't immediately turn that away. It's important to try things out. So if you don't know, you can always try a simple germination test. Preferably, the industry standard is 200 seeds, but for at-home use or for smaller scale use, you could test with anywhere between 50 to 100 seeds, just depending on how much you have available to you. And just conduct a simple test. Soak those seeds in water overnight. Place them in a paper towel or Ziploc bag, in a paper towel and a Ziploc bag, keep them in the sunlight or expose them to indoor UV light at room temperature, 70 to 80 degree Fahrenheit. And get children to do this. Kids love working with seeds, and this is a really great hands-on way to include different generations in this type of research. And then the general rule of thumb is to give your seeds about 4 weeks. Most milkweed will germinate way sooner than that, but that's kind of like a generous time frame to give yourself. And after that, if you're not getting good germination, you either have a dormant seed or you have maybe some low viability. So when you are starting your germination, always I recommend storing milkweed, and this will apply for a lot of native plants as well. Just storing those seeds in cool and dry conditions, whether that's a paper bag or pill bottles, and keeping them in your fridge or freezer. Even for non-dormant seeds, having that transition, that temperature transition, kind of just helps them get into gear and germinate a little bit faster. And then when you are ready to germinate, exposing those seeds to light and fluctuating day and night temperature as well. Basically, you're mimicking that natural condition that it might be facing in the wild, as well as keeping moisture consistent. So if you're putting these seeds on potting medium, paper towel, whatever you're using, checking daily as much as you can, because if seeds dry out, you might lose some of that germination success. And then what I found and what's pretty common across the board for milkweed germination is you should get most of your germination occurring between 1 to 2 weeks. And some I found even reached 100% within 3 days. So if your germination is getting low percentages, and I'm so sorry, I'm going to be moving and walking because I need to plug in my laptop. My house currently doesn't have heating, so I was in the warmest house or warmest room in the house, and I didn't have my charger with me. So okay, we're plugged in, we're good. So getting your seeds tested for viability, this is not something that everybody has the budget for. So you can actually do some of this in-house. There's very simple methods. First and foremost, I would just carefully inspect your seeds. You can actually tell if there's an embryo in there. You can either squeeze it and it should have a little hard ball in there, essentially. If it's completely flat, it's likely that there is no embryo in there. And you can actually cut those seeds open yourself and stain them with a tetrazolium chloride solution that you can buy commercially available. Um, Essentially what you do when you stain those seeds, there's a whole protocol that I can if you email me, I can send it to you. But if you literally just Google TZ protocol, there's a lot of at-home recommendations and articles that kind of outline this really well. But basically, you're just exposing your seed to a chemical that, when it interacts with hydrogen, it stains the embryo red. And so, if your seed is alive, if it's viable, it's constantly respiring, and so there's that process involves the hydrogen, and then if it interacts with the tetrazolium, then you'll get a red stained seed. That's essentially how it works. You can also get viability percentages from the grower. They should provide this to you. In my experience, that was not always the case. So you might have to do at-home testing if you have the funds for it, or if you have great relationships with other seed labs. I utilize the Colorado Seed Lab in addition to doing at-home testing. They will actually do the testing for you. The only downside is that oftentimes they require a huge quantity of seeds to really get a good, accurate picture of how viable your seeds are. Usually that's 200 seeds, so quite a lot of seed if you don't have a big selection. And then if germination is low, even if you have good viability and you've encountered dormancy, there is something called the move along experiment, outlined by these two fantastic researchers, Carol and Jerry Baskin. They're like the seed, I don't know, they're like all knowing when it comes to seeds. They spent their whole professional lives studying seeds. And this research project was sort of designed after a move along experiment. But if you look up that particular title, it kind of helps you like kill two birds with one stone as you're pushing seeds through different temperatures to figure out what's going to break their dormancy. Um, if you do have dormant seeds, I recommend rinsing the seeds in bleach and water solution before putting them in your paper towel or your vermiculite or your sand. I do recommend paper towel just because it's easier to keep track of those seeds and actually inspect them for mold, because you will get mold. You're going to reduce the mold with bleach, but you're not going to completely eradicate it. It's pretty much impossible to get rid of all microorganisms on seeds and in anything that you're working with. It's on your hands, it's everywhere. So placing those seeds on the paper towels, making sure they have their own space. Don't fold them on top of each other, because that also encourages mold. And then putting them in the fridge. I actually recommend doing a 4-week stratification and seeing if that works first. I mistakenly did 6 weeks, which I think was a little too long and encouraged a lot more mold growth than I would have otherwise had without, if I did the 4-week. But I think anywhere in that time window will be sufficient based on our results and results from other studies. And I would actually check every week for mold. If you do encounter mold, no problem. Take the seed out. You can actually just rinse it and wipe it off. If it's like mushy and gross, it's just been completely consumed by the mold, throw it away and start a new paper towel. It's a little tedious, but it will save you losing all your seeds to mold, which you don't want to do. You don't want to waste those seeds. And then again, with the cold stratification period, I would actually start with the baseline of 4 weeks, but you can always have extra in your fridge and have them linger for another couple weeks, just in case something were to happen to that 4-week batch. It's just important to have like a backup plan, because you don't want to put, all your seeds all at the same time, all at once, because sometimes things happen. Irrigation can break, or yeah, again, mold can happen or fungus can grow. So it's good just to have a backup plan for that. For non-dormant seeds, because those seeds that were dormant were going through those stratification, that whole time, those seeds are able to imbibe some of that water from the paper towel, that moistened paper towel. If you're working with non-dormant seeds, a great way to enhance germination is just to soak them overnight, because again, you're not putting them through that cold stratification, so they're not imbibing water. Unless you put them in a bowl or a cup of water. That is a great method. Desert Seed Resource Center, which I'll link at the next slide or one of my later slides, also has a lot of success with this protocol for non-dormant seeds. Please record or keep track of your germination data, even if it didn't work. I think a lot of time in academic literature, like nobody wants to publish failures, but we need to know about the failures because we don't want to repeat things. We don't want to repeat mistakes. We can learn from those mistakes. And that type of data is still really useful for us and for sharing with your community who's interested in growing germination, who's interested in germinating milkweed. So please record on spreadsheet, Google Sheets, whatever you have. It's really important to keep track of that. And then I think this is my last slide. Some of the really fantastic folks that I encountered, and this is not a comprehensive list by any means, but just from my experience, I wanted to provide this list if any of you want to take a photo of this, or I guess the slides will be shared. Happy to share this. Getting milkweed seeds. Believe it or not, some of the companies that I encountered did not know where their milkweed came from, or they straight up lied. And I'm sure it was unintentional, but in my opinion, that is not acceptable, especially if you're working with native plants. It's really important to know where your seeds are coming from. There was a vendor that was selling seeds from Australia with no regulation. A lot of forbs, I think, if not all, wildflower specimens are not regulated in the same way that grass specimens are in terms of the USDA and how seeds are shipped. So you have to be on top of it. You have to do the research. You have to make sure that you know where these seeds are coming from. Places that were really on top of that were local botanical gardens. Oftentimes they will collect from the wild, or they'll have some seeds stored, and they're happy to share if they have enough to share. Your local native plant societies will often have growers or collectors. Monarch advocacy organizations, of course, because they're interested in working with milkweed, they'll often have recommendations or even a milkweed seed to give away. A company called Grow Milkweed Plants, which is run by Brad Grimm, really great resource. He does a lot of germination testing as well, and so he can give you recommendations. And he has seeds from all over the country. Western native seed, that's more for us in Arizona and throughout the Western US, Colorado and New Mexico. They're a really great resource. They do wild collected. Borderlands, exact same thing, wild collected. They are starting to do their grow out program, so they should be having more milkweed available from the Patagonia area in Arizona. Prairie Moon Nursery, that's more Midwestern, but they have a really great record keeping of where all their seeds are coming from. Ernst Seeds, Everwild Seeds, these have a lot of a wide range. Although I think Ernst is more confined to the Northeast, US. Conservation Blueprint, Native American Seed, those are all companies that I collaborated with or got seeds from. Some other great resources that just have general information on milkweed germination are the Xerces Society, Desert Seed Resource Center, which is more local here in Arizona, and then the Lady Bird Johnson Wildflower Center. But please, by any means, if y'all have resources that you want to throw, just to put them in the chat, because this slide is by no means comprehensive. And last but not least, I just want to give credit to those folks who did help me with this research. I have a long list of people. I just wanted to throw their names out there. I was able to get free seeds from the Desert Botanical Garden here in Arizona, as well as Tohono Chul Park. Bamrit Seed donated some seeds, Idaho Botanic Garden. My advisors, Elise Gornish, who's moderating this panel, as well as Dr. Jeff Femi and Rachel Mitchell. And then, of course, folks who donated seeds and helped just provide great advice throughout this process. So I will end it there and leave it for questions. I hope I didn't go too much over.
You didn't go over at all. You're exactly on time, Philippa. Thank you so much. Okay, I'm going to start rolling back these questions. So Sarah asks, how long do you recommend before you plant milkweed in the ground? 1 gallon pot size, question mark?
Say that first part again.
How long do you recommend before you plant milkweed in the ground? And Sarah, feel free to elaborate on your question in the chat.
Yeah, so this will not be supported by evidence from this particular research because I didn't plant, but when I was working as a conservation horticulturist and we did do milkweed transplants, we found that milkweed were very easily vulnerable to predation. I think a lot of insects really like milkweed and want to just eat it up. So I would actually recommend like a robust 1 gallon plant before you send it off into the wild. I know a lot of people even encounter like aphid problems in even in the greenhouse if you're doing greenhouse production, which we also encountered, and we sprayed organic pesticide in the greenhouse. So it was controlled. We weren't like spraying, out in the field. But yeah, that I would recommend that. I will say though, don't give up on milkweed if it disappears. We often encountered like the stems were stripped bare, but their roots are pretty robust, and I would wait until you see a new leaf pop up before you decide if that plant is no longer alive. But yeah, I think a 1 gallon, a nice robust 1 gallon plant would be more ready for the elements, ready to battle whatever bugs are coming at it.
Awesome. Have you found a rough estimate of viability in milkweed in years or months?
Not really. I encountered a couple of seeds that I could not include in the study because they were like lower than 50% viability. Some were even 20%. I'm not sure it's worth like germinating those seeds. I mean, you could try, but I wouldn't like put all your resources into that. I didn't find any particular pattern, but generally speaking, for native plants, wild harvested plants typically have a lot more um, variability in their viability than a commercially grown. Mainly because if you're growing in a farm setting, you're controlling the irrigation, you're probably managing for pests. So you're kind of giving those plants everything that they need. Not always the case, but I would say if you're really concerned about viability, go with a commercial grower and ask them what that percentage is. And they typically won't sell you seed unless it's got uh, high integrity. Although always important to ask because I was sold like 20% viable seed. So yeah, sorry, I don't know if that answered your question, but
Same. Um, Someone asked if you have any measurements for East Coast milkweed variations.
yes, some of those populations were, when you say measurements, I'm sorry, are you talking about like germination or
It just says measurements, TEA, if you want to chart, maybe
yes.
this, hopefully, so maybe the germination, they mentioned a chart. Yeah.
If you, okay, if you're working with the Northeast, chances are, so common milkweed has been pushed as like the big, like if you're gonna do milkweed, do Asclepias syriaca. I will warn you that it propagates through rhizomes, so it will take over. And somebody mentioned that in their chat that they noticed an aggressive species. So that could be a good thing or a bad thing depending on your landscape goals or your habitat goals. But for that particular species, there are not just dormant populations. I actually found a lot of non-dormant populations. They are not reflected in this particular graph, but I would be a little more scrutinous about common milkweed, making sure you know what the dormancy is. But those typically give pretty decent germination, and from what I found, above optimum levels in both of those temperatures. So they seem to be pretty flexible, but I would be more careful about the dormancy. Same thing for Asclepias incarnata. Swamp milkweed is notoriously finicky for germination temperature. Um, and I would say tuberosa, I should have had more in this graph, but that for butterfly milkweed tuberosa, that is a pretty commonly grown one, and it found in almost like every nursery that sells milkweed. It germinates pretty well, regardless of stratification or temperature. However, the lower latitude populations I did not have great success with. So I would actually stick to the higher latitudes, maybe some plants that are grown in the Northeast. Um, yeah, I don't know if that answers your question, but I would say if your milkweeds are coming from the Northeast, they'll probably have um, some level of dormancy just because of those seasonal changes. And Ernst Seed is up in Pennsylvania, and they were a really great resource for milkweed for that particular region.
Awesome. Thank you, Philippa. Okay, everyone, if you have other questions for Philippa, you can send her an email. I put her email address in the chat. And right now, we're going to give everyone time for some lunch. So we are going to take a pause between now and 12:15. And to try to keep us on schedule, I'm gonna start us like on time at 12:15. So you can stay on or you can